are required for the plant cell growth. After that, the organic compounds. When we consider organic compounds, in case of organic compounds, you will see that the carbon sources are nothing but the sucrose molecules. And when we consider sucrose molecules, these sucrose are needed as a carbon sources. After that, when we consider vitamins, we require adenine, inositol, as well as thymine for the different types of the cellular activities when we consider the plant cell. Also, we always require another different types of the organic materials such as the citric acid, which is always required, or it can also be called as ascorbic acid. And when we consider this type of the antioxidants, they are major part of the Krebs cycle. As well as we also require some phenolic compounds, just like the fluoroglucinol, which stimulates the rooting of the shoot sections or shoot subcultures. And when we take that shoot subcultures, phenolic compound generally stimulate the rooting. Also, we can add different types of the natural complexes in our tissue culture media. That is the coconut endosperm, fish emulsion, after that protein hydrolysates, as well as tomato juice, yeast extract, malt extract, potato agar, etc. So these are the element complexes which are always required in or which are always taken from the natural structures. After that, when we consider the charcoal, activated charcoal is always used as a detoxifying agent, because when we consider charcoal, it always show absorption of the different types of the impurities. And when we consider the charcoal, it always show the detoxification of the tissues or our culture. After that, when we consider the plant tissue culture or our tissues, the tissues always enhanced by the different types of the plant growth regulators. And when we consider plant growth regulators, there are different types of the plant growth regulators, such as when we consider the plant growth regulators, auxin always induces the roots. When we consider cytokinin, they always induces the shoot systems or shoots. When we consider gibberellic acid, they always show, or they are always responsible for the cell enlargement. When we consider abscisic acid, it can be used as a plant stress hormone. And when we consider that abscisic acid, even though it is a stress hormone, it is very helpful for the stunted growth of the your plantlets inside the tissue culture medium. But when we consider the growth regulator ethylene, if it is produced in larger quantity in our tubes, this type of the ethylene can be harmful. So ethylene is bad here for our tissue culture. And for that purpose, we require always ventilation for our cultures. Now, one by one, when we consider auxins, auxins firstly used for the rooting, and along with rooting, we can also obtain the callus as well as the embryos uh, by the use of auxins. So, what are the examples of these auxins? They are IAA, IBA, NAA, 24D, 245T, as well as the picloram. So, these are some of the examples of auxins which are commonly used in our tissue culture media. After that, when we consider cytokinins, cytokinins always enhances the shoot formations in case of the plant cells. So when we go for the tissue culture, if we add cytokinins in somewhat larger quantity, actually larger quantity is, it is always in parts per million or ppm. And when we add these cytokinins in larger quantity than the auxins, then we always show the, or we always observe the formation of the shoots. And when we consider examples, benzylaminopirin or BA, 2IP, kinetin, zeatin, these are some of the examples of commonly used cytokinins. After that, when we consider gibberellins, actually they are not that much used in tissue culture media. But when we consider separation of root formation and formation of the adventitious embryos, we can use the gibberellin. As well as when we consider the growth of meristematic tissue or growth of the axillary buds, and we have to take the tissues disease free, in that cases you can directly use the gibberellin for a rapid growth of the apical buds. When we consider abscisic acid, it is generally not used, but when we consider adventitious embryo formation or embryogenesis, you can use abscisic acid. After that, as I earlier discussed, ethylene is a bad. When it is always present in larger quantity, it suppresses as well as it enhances the senescence, that is the falling of the leaves as well as the falling of the stem part, or generally the falling of the flower or fruiting part if they are present inside the tissue culture media. When we consider hormone combinations, we have to make different types of the hormone combinations. We cannot use auxin only, we cannot use cytokinin only, we cannot use only gibberellic acid, we cannot use only the abscisic acid. So we have to make hormone combinations. For callus development there is a different combination, adventitious embryogenesis there is a different combination, for rooting of shoot cutting there is a different combination, and so on. So when we consider these tissue culture media, and we always require the support system. Why we require support system? Because when we consider the plant tissues, when they are embedded inside the water, in our house also, when we take different types of the vegetables and when they are kept in water for longer period, what will happen? You will observe that when we consider these type of the vegetables which were kept in water for longer period, they generally show the decaying. And when we put our tissues also inside the media, we also require the support system for our these tissues, otherwise it will again show the decaying of the our tissues. So commonly used support systems are nothing but agar, and when we consider that agar, it is obtained from the seaweeds, as well as we can use the agarose, as well as we can use the gelrite. You can use different types of the mixtures, as well as we can use the mechanical structures such as the bridges or rafts which are prepared from your papers, as well as we can use the sand. When I was working in VSI, Vasantdada Sugar Institute, Manjri, our guide, Mr. SG Dalvi, he has developed another one support system method, and it was simply using the cotton. So this type of the support systems are required for our callus as well as the different types of the tissues for proper growth. After that, when we consider media formulations, depending upon culture, we always show, or we have to develop different types of the media formulations. We have to adopt the different types of the media formulations. So there are numerous. When you search a website or Google it, you can observe that there are numerous media formulations. We have to differ different types of the salt concentrations. We have to also differ the hormone concentrations. And when we consider common tissue culture media, it is nothing but the MS media. As I discussed last day, it was prepared by Murashige and Skoog in 1962. So these are the different types of the media which are used or which can be formulated according to our requirement. So after that, the main concept or main principle regarding plant tissue culture is the sterilization. Without sterilization, you cannot go for the plant tissue culture, and tissue culture always require the aseptic conditions. So what is mean by sterilization? Sterilization, it is nothing but the killing or excluding microorganisms or their spores with heat, filters, chemicals, or other sterilant. By using any method, you have to just get rid of the different types of the microorganism, and it can be just done with the help of the sterilization techniques such as the heating, filtrations, using chemicals, as well as different types of the sterilization techniques. They can be used for the sterilization. So when we consider aseptic techniques, when we use aseptic techniques, we generally obtain the sterile environment. It is always free of pathogenic microorganism. It is free or freed from pathogenic microorganisms. It is always free from the living germ of diseases. So these are the common terms. So when we consider aseptic condition, it is always involving that we have to just kill or exclude the microorganisms which are not required for our tissue growth. After that, the culture, when we consider axenic culture, which is a very pure culture, and when we consider that culture, it is always germ free, It is uncontaminated, that is no growth of the any pathogenic organism. If after that, when we consider not only pathogenic organism, you cannot see any other or microorganism when we consider our axenic culture. It contains only the single plant tissue, that is the single organism only. It is not showing another different types of the tissues or organisms which are present in our culture. Then that culture is called as a axenic type of the culture. So what are the different types of the sources which always show the contamination of our tissue culture? So it may be simply the explant or culture. When we directly take explant from the soil, it may cause the numerous type of the contaminations of different types of the bacteria, fungi, as well as the different types of the other microorganisms. So first of all, we have to go for the sterilization of these explant, and after that, when we consider another source, it is nothing but the vessels, the glasswares, as well as the different types of the other equipments, smaller cutters, as well as the different types of the forceps. They can be the source of contamination. So first of all, we have to again they sterilize them. After that, the media. When we prepare a media, we have to always make combinations of media from one container to another. We have to use water, we have to use different types of the other solutions. And when we consider these solutions and when we formulate media, this media also should be sterilized, so it can be a source of contamination. After that, the instruments. So when we consider different types of the instruments used for the tissue culture media, these also must be sterilized. And the environment where handling is taking place, that is the our culture room, as well as transfer room, as well as the media preparation rooms, they must be always the sterilized, or it must be a contamination free. So when we consider aseptic techniques, these techniques are generally of two different types. First one are called as a chemical, and second one are called as a physical. When we consider chemical techniques, in chemical techniques we have to use the disinfectants. After that, different types of the antibiotics, as well as the subliminary compounds. When we consider the physical treatments, in case of physical treatments, we have to simply go for the heating, or we can also go for the electromagnetic radiations, we can also go for the filtration, and we can also go for the ultrasonic waves. So these are the different types of the aseptic techniques can be used, which can be used for the sterilization. So when we consider disinfectants, when we consider these disinfectants, they can show the following different types of the activities. They generally penetrate into the bacteria. Also, they will denature the bacterial proteins. They decrease the activity of bacterial enzymes. They inhibit the bacterial growth as well as the metabolism. They damage the structure of cell membrane, and they can change the membrane permeability. And if this happens, you can directly see your culture, which is always a bacteria free as well as the virus free. So these type of the disinfectants can be used for our media. After that, when we consider disinfectants, these different types of the disinfectants can be used for the surface sterilization, such as liquid laundry bleach, that is commonly used as commonly used compound, that is NaOCl, sodium hypochloride, as well as we have to use sterilized waters, calcium hypochloride, ethanol, as well as we can use the different types of the alcohols for our sterilizations. When we consider antibiotics, the antibiotics such as the streptocycline, carbenicillin, cefotaxime, rifampicin, these type of the compounds, antibiotics or compounds can be used as a disinfectants or sterilizing agents. When we consider antibiotic, which antibiotic should be used? So when we consider that ideal antibiotic, it must show broad spectrum activity. It should not be having the resistance for that particular bacteria or virus. After that, when we consider these type of the antibiotics, they must be have the selective toxicity. And when we consider these type of the antibiotics, they must preserve the normal microbial flora of that culture media. Also, we can go for the UV radiations. When we consider UV radiations, when we induce the cell culture or tissues more than 2 minutes or 3 minutes, you can get the sterilization. After that, also we can obtain the sterilization by heating methods where we can simply use the ovens, you can simply use the microwaves, you can simply use the different types of the flaming or heating tools. Also, the major instrument which is used for the heating, it is nothing but the autoclave. And as a student of biotechnology, you are very well known about this autoclave. We have to just keep the pressure at 15 lbs, and we have to get the temperature near about 120 degrees Celsius, and that can be always used for as a standard procedure for our autoclaving. After that, when we consider flaming or heating tools, they are also called as a bacticinators. And when we consider this type of the cinators, they also show the presence of smaller glass beads. And when we consider these glass beads, it can be heated simply with the electric current, and we have to just keep our tools inside that glass beads. Also, if you have to use the heat labile compounds or heat labile substances, then you have to simply go for the filtration. When we use the GA, when we use the IA, or when you have to use the another different types of the proteins or antibiotics, then you have to just to go for the filtration with the simple cellulose filters. And these type of the filters are always used for the sterilization of heat labeled compound, which cannot be simply autoclaved. Also, the surrounding area or surrounding air, which is always should be filtered, and it is always obtained by the HEPA filters, which are always attached to the larger laboratories. So when we consider autoclaves, you have just observed that autoclaves, which is up to size of near about 3 to 4 feet, and you can just simply this type of the autoclaves, which are just long enough, as well as the height of these autoclaves, which is kept horizontally, height is just more than us. So these type of the larger equipments which are used in tissue culture techniques. So as I just discussed earlier, so these are the glass beads, which where we can directly put our scalpels, as well as the forceps, as well as you can use the flaming, as well as alcohol. So, and this is the uh, at laminar airflow. And when we consider this type of the laminar airflow, they generally show the presence of HEPA filters. As I earlier discussed, when we have to go for the filtration of the heat labile compounds, you have to just use this type of the simple filters, which are called as cellulose type of the filters, and you have to just inject those liquids inside your media. So just now to this, I'd like to conclude my small talk. हा थांबव ना म्हणजे हा चालेल चालेल हा म्हणजे मी हे केलं हा थांबवलं म्हणलं मी आता हा चालेल चालेल